right, you guys, welcome back. And today we're actually going to be starting our next chapter in biology, which is chapter 3.3, Genetics and Heredity. We're going to begin this by first talking about something called meiosis. So meiosis, as we're going to talk about, is another form of cell division similar to mitosis, but definitely not the same thing. All right, so let's get going and get started. If we're going to talk about meiosis, first we have to talk about something called sexual reproduction. Now, in our previous chapter, we mentioned asexual reproduction, which is when one parent gives rise to an offspring that is genetically identical to it. However, with sexual reproduction, you actually have a genetic exchange of material between two parents. So you get a little bit of DNA from one parent and a little bit of DNA from another parent, and this allows for genetically unique offspring. When we mean genetically unique, yes, they have genetics in common with both parents and even with siblings, but they're not going to have all of their DNA identical. The only time that occurs is either asexual reproduction or identical twins in sexual reproduction, but that's a whole other thing. So you have two genetic sources, what we call parents, and they combine their DNA together in order to get this genetically unique offspring. So to do this, we have these particular cells called gametes, also known as sex cells. Now, these are very, very special cells. All cells in your body, with the exception of one, are considered somatic cells. Somatic meaning body, and that means that they have the full DNA set. But these gametes, they're the only cells in your body that do not have the full DNA set. We'll talk a little bit more about what that means in a moment. But they are very, very unique cells, and their sole goal is for sexual reproduction. That's the whole point of them, which is why we also do call them sex cells. And in males, we consider them sperm. We also in plants kind of think of it as pollen, but in one of my videos, I explain why that's not exactly accurate. Pollen holds the sperm, but pollen itself is not the sperm. And females, we consider these to be the egg cells. As you can see in the picture, we have, if you follow the mouse, we have the egg cell, we have the sperm trying to get in, and then the head of one sperm will actually do what's called fertilization, which means that the male gamete, in this case the sperm, fuses its head with the egg. And this allows it to contribute its genetic information. And then the two, again, looking at the picture, the male pronuclei and the female pronuclei, they will come together to form one nucleus within the egg. We call this fertilized egg cell a zygote. So fertilization is simply the gametes coming together to form a fertilized egg. So now we have half of the information in that nucleus from mom and half of it from dad. As we've mentioned in the past, your body cells and most of your cells, again, except gametes, are diploid. Diploid meaning that they have the full set of genetic information. Humans have 46 total chromosomes. And it's like that because we actually have 23 pairs. So we have 2 copies of each of our chromosomes, and we get one from mom and one from dad. That's how we get our pairs of chromosomes. So somatic cells are diploid. There are 46 total. They come in 23 pairs. But the gametes, on the other hand, they have half the number of chromosomes. And what we mean by that is instead of having 2 copies of each chromosome, then they actually only have 1 copy. So that means that there's only 23 chromosomes in an egg cell and 23 chromosomes in a sperm cell. That's what it means to be haploid. A lot of times we can also delineate this with the letter N, as you see right here on the screen. And with diploid, we call that being 2N because we have 2 copies of all of our chromosomes. So if sperm is haploid, meaning it's N, and an egg is haploid, meaning it's N, we have N plus N equals our fertilized egg, the zygote, as 2N. So in other words, 23 chromosomes from dad's sperm plus 23 chromosomes from mom's egg come together. We now have 2 copies of every chromosome, giving us 23 pairs or 46 total chromosomes. You mentioned that meiosis is a type of cell division, and what we mean by that is that you start off with one cell and you're going to get multiple cells as a product. However, instead of giving rise to more somatic cells like mitosis does, this is actually going to give rise to the gamete cells. Both mitosis and meiosis are types of cell division, but there are some key differences between them. First of all, mitosis is one set of division and it has certain products, certain daughter cells that we're going to mention in a minute. Meiosis, though, is actually 2 sets of division and has very different product cells. So in mitosis, we go through one set of division and that we start off with our diploid cell. It goes through division. We end up with 2 daughter cells, both of which are also diploid. Not only that, but they are genetically identical to each other and genetically identical to the original parent cell. So the point of mitosis is simply to make more of the exact same, as opposed to meiosis. Meiosis is actually what we call a reduction division. And that kind of sounds a little weird at first because, well, we actually make 4 cells out of it, so it sounds like we're making more. But when we talk about reduction, we're actually talking about the number of chromosomes. So when you have meiosis going on, you start off with 1 diploid cell, but then after it goes through both sets of divisions, you end up with 4 daughter cells that are each haploid. So we are reducing the number of chromosomes in each of the daughter cells, giving us less chromosomes. This way, when they fuse with another gamete, we can make a full diploid cell. All right, let's start talking about the process now, shall we? As we said, there's 2 types of 2 sets of division in meiosis. We simply call them meiosis 1 and meiosis 2. So, sorry, in the first set of divisions, in meiosis 1, we take the homologous pairs of chromosomes. Remember, homologous pairs are the 2 of the same chromosomes. So you have 2 chromosome number 1s. They have all the same genes on them, but they might have different alleles, which are variations of those genes. But you have 2 of each chromosome, and that's a homologous pair. And in meiosis 1, we're going to take that homologous pair and separate them so that you no longer have homologous pairs in the daughter cells. Since there are no longer pairs of chromosomes in the daughter cells, we separate the homologous pairs. Then the result of meiosis 1 is going to be 2 haploid daughter cells. Well, let me show you a picture. That's just kind of a bit or a brief overview of it. We're going to go through each one of these steps. But before we do that, we got to talk about interphase 1. We learned about interphase when we talked about mitosis. And the key thing here, just like then, is that during S phase of interphase 1, we replicate the DNA. So we copied all of the DNA so that we have more to put into the cells. Don't forget, in meiosis, we're not going to have just 2 daughter cells. We're actually going to have 4. So we do go through a regular interphase beforehand. And since it occurs before meiosis 1, we call it interphase 1. Prophase 1 is going to start off kind of similar to prophase in mitosis. The nuclear membrane disappears, the chromosomes become invisible, meaning they go from the chromatin form all the way to the chromosome form. The centrioles are going to start moving, and the spindle fibers are going to start forming as well. They're being built by the centrioles. But something different here is that what we're going to see is the homologous pairs are going to get super duper duper close together. So the homologous pairs with their duplicated sister chromatids get super duper duper close to the point where they actually fuse and touch and fuse, sorry. When they get super close together, we call that a tetrad. And then this particular event called crossing over is going to occur. Let's look at more detail at that before we move on. So crossing over is when, as you can see in these pictures here, picture 1, we have our homologous pair. They get super duper close. When they get super duper close like that, we call it a tetrad because there are 4 sister chromatids in those 2 chromosomes. Now, if you notice in picture 2, they're so close right here that they can actually overlap. When they overlap like that, something called synapsis is going to happen. That means that they're actually going to fuse together. And then when they separate, if you notice in picture 3, we now have exchanged some of the alleles. Now, we don't have different letters, we just have different variations of those letters, which means that we have new combinations of alleles for the exact same genes. So we're not adding new information, we're just changing the type of information. We still have A through E on all of the chromosome or sister chromatids, but now we just have new combinations of capital versus lowercase. So this is what crossing over is. All right, so now that crossing over has occurred, we have metaphase 1, which is the centrioles are now on opposite sides of the cell. They built their spindle fibers, and the spindle fibers are attached to the homologous chromosomes, and they've lined them up in the middle. But they've lined them up as pairs, so 2 by 2. They've lined them up as pairs 2 by 2 in the middle of the cell. That takes us to anaphase 1. The centrioles pull on the spindle fibers. That's going to cause the spindle fibers to pull on those homologous chromosomes, separating the pairs to opposite ends of the cell. Once the pairs have been dragged all the way to the opposite ends of the cell, we have telophase 1, which is where the nuclear membrane is going to reform around the chromosomes. They're going to temporarily go back into chromatin form. The spindle fibers will break down, and cytokinesis is going to occur. Cytokinesis is when the cell membrane starts pinching inward in the middle, and that's going to cause the organelles and the cytoplasm to stream to the 2 ends. Once the cell membrane fuses during cytokinesis or meets in the middle, it's going to fuse, and then the 2 daughter cells are going to pop apart. Now remember, they are haploid because we separated the homologous chromosomes. We no longer have homologous chromosomes in our cells. So we are no homologous chromosomes means no longer diploid, so we now have 2 haploid daughter cells. And that's going to bring us to meiosis 2. Now, before we start talking about this, there is no true interphase between meiosis 1 and meiosis 2. There could be a rest phase, yes. And what I mean by a rest phase is the cell can use this time to check, make sure everything went through the first set of divisions correctly, and then it can gather more material to build new spindle fibers. It can gather more glucose to provide energy for the next set of divisions. But there is no true interface. Why do I say this? Because we do not duplicate our DNA. A key part of interphase is duplicating the DNA. We don't want to do that yet. We still have our previous duplicated sister chromatids attached. We're not going to make more copies of them. So we are not going to duplicate our chromosomes. Therefore, it is not a true interphase. All right, so that brings us to meiosis 2. And the 2 cells that we ended up with in the previous meiosis 1 division are both going to enter into meiosis 2. And that's why you see 2 lines of division here, because we had these 2 cells entering post meiosis 1. So 2 cells end in meiosis 1, 2 cells will begin meiosis 2. And this time we're actually going to separate the sister chromatids at the centromere. Remember, that's where the sister chromatids are connected. And we're going to separate them so that we no longer have our duplicated buddies with us. All right, so first part of meiosis 2 is going to be prophase 2. A lot of meiosis 2 is going to be similar to mitosis. Nuclear membrane disappears, the centrioles start to move, the spindle fibers start to form, the chromosomes become visible, meaning they go from the chromatin form to the chromosome form. They're going to start moving around. And that's really all that's going on in prophase 2. There is no crossing over at this point because crossing over occurs between homologous pairs. We no longer have homologous pairs, so that means that we are no longer able to do crossing over. That was a strictly prophase 1 thing. Then we have metaphase 2. The spindle fibers attach to the chromosomes and help line them up in the middle of the cell. So they are lined up in the middle, single file, just like what we saw in mitosis. And then the centrioles are going to pull on the spindle fibers, causing the spindle fibers to pull on the centromeres, and that's actually going to cause the centromeres, which are holding the sister chromatids together, to separate. And that splits the sister chromatids and allows the spindle fibers to start pulling them to opposite ends of the cell towards the centrioles. From there, we have telophase 2. The nuclear membrane is going to reform around the different groups of chromosomes. The spindle fibers break down. The chromosomes go from chromosome format back into chromatin format, and then cytokinesis occurs. So once again, the cell membrane starts pinching inward. As it does, cytoplasm and organelles stream to the two ends. The cell membrane will fuse in the middle during cytokinesis, and then they will separate into cells. And we are left with 4 haploid, genetically unique daughter cells. So let's take a look at the full diagram for meiosis, and that's what we see here. So if you follow the movement of my mouse, here we have interphase 1, looks just like a normal cell, and we duplicate our DNA at that point. In prophase 1, the nuclear membrane has disappeared, the centrioles are beginning to move and build their spindle fibers, and the tetrads have formed. So the homologous pairs have gotten super duper close together that they actually can overlap, fuse, and exchange information. So we had crossing over occur. Then here in metaphase 1, the chromosomes are attached to the spindle fibers, and the spindle fibers move them to the middle of the cell and line them up as homologous pairs. Notice that they are 2 by 2. Anaphase 1, we have the spindle fibers pulling on the chromosomes and separating the homologous pairs to different sides. So the red and green will be pulled up. The yellow and blue are going to be pulled down. That takes us to telophase 1. Notice that the nuclear membrane is starting to reform around each group of chromosomes. Spindle fibers have been broken down, and the chromosomes will actually turn back into chromatin format for a break, so that we can gather material and check to make sure everything is fine. And that takes us to meiosis 2. So again, you can see the end product of telophase 1 is going to be 2 haploid cells. Both those cells enter meiosis 2. So we have both of the cells, the nuclear membranes are gone, the spindle fibers are starting to form, the centrioles are starting to move. In metaphase 2, the spindle fibers attach to the chromosomes and line them up single file along the center of the cell. And then here in anaphase 2, we see that the spindle fibers are pulling on the sister chromatids, separating the sister chromatids and dragging them to the opposite ends of the cell. And then telophase 2 in cytokinesis, nuclear membrane reforms, spindle fiber breaks apart, chromosomes go back in chromatin form, and then they pop apart. So we now have 4 haploid, genetically unique daughter cells as a result. All right, so now that we kind of looked and talked about what meiosis is, let's do a comparison between a mitosis diagram and meiosis diagram. Again, so we can just kind of do a side-by-side comparison in order to figure out and know the differences between them. So first, we begin with our diploid cell there in the middle. It is diploid. It has two homologous pairs, the big red pair and the small blue pair. You'll notice that the red has different shades of red and the blue has different shades of blue. That's more so we can tell, that they came from, that they are the same chromosome, but two different variations of it. So in mitosis, which is simply a way to make more of the exact same cells, we have our 4 chromosome or 2 homologous pair cell begin an interphase. It's going to duplicate its DNA and then go to prophase. Spindle fibers form, nuclear membrane starts to disappear, the centrioles are moving, and we can see the chromosomes. They are visible, meaning they switch from chromatin to chromosome form. In metaphase, the spindle fibers have attached to the centromere of the cell, of the chromosomes, and line them up in the center of the cell, nice and single file. In anaphase, the spindle fibers are going to pull on the centromere, which is going to cause these sister chromatids to separate and be pulled to opposite ends of the cell. And then telophase plus cytokinesis, we are left with 2 daughter cells that have the full set of DNA. And they even show the chromosomes underneath. So we have 2 red and 2 blue in each one of these. Well, we began with 2 red and 2 blue. So we still have homologous pairs, we're still diploid, and it's exactly the same to each other. So both daughter cells are genetically identical, and they're genetically identical to the original cell too. Now let's compare this with meiosis. Again, start off with our diploid cell. We're gonna replicate our DNA in meiosis 1, and then we're gonna go to prophase 1. The nuclear membrane's disappearing, spindle fibers are being formed, centrioles are starting to move, but we see that the homologous pairs get super duper close. They're forming those tetrads, as you can see here, to allow for crossing over to occur. So they get so close, they overlap, fuse, and exchange information. We now have one of the dark red sister chromatids with a little bit of light red on it, and one of the light red sister chromatids with a little bit of dark red. Metaphase 1, the chromosomes line up in pairs in the center of the cell. And then in anaphase 1, we separate those pairs to opposite ends of the cell. Telophase 1, we separate the cell. We now have 2 haploid cells, haploid because they no longer have homologous pairs. And that takes us to prophase 2. So nuclear membrane is going to disappear. We're going to, again, have in metaphase 2, the spindle fibers will connect up and attach to the sister to the chromosomes at the centromere. Anaphase 2, we're going to separate the sister chromatids, and telophase 2, we can see the products down here. We have 4 daughter cells, not 2, but 4. And if you notice, each one only has 1 red and 1 blue chromosome. So we went from having 2 reds and 2 blues to 1 red and 1 blue, 1 of each, which means we do have haploid cells. Not only that, but none of the color combinations are identical. We have all dark red with all dark blue. We have dark red with a little bit of light red, dark blue. We have light red with a little bit of dark red, light blue. We have all light red, all light blue. So we can see different combinations of alleles for those different respective genes. And that's what allows for genetic variation. Now imagine that the blue pair also went through crossing over. And imagine there's 23 pairs of chromosomes instead of just 2. There's a lot of variation that can occur. All right, so we do have some additional resources. I'm not necessarily going to go into and have us watch them together in this presentation, but I would recommend that you go and watch them once you're done. Consider it part of the lesson. And what you should do is I will post the link to this Prezi in the description so you can access it and thus access the links to the different videos. So what we have 3 videos done by me in my Science in Pajamas YouTube channel. The first one looks at meiosis and kind of goes through a review of how meiosis occurs. I'm not going to watch it now. Then we have the next one on chromosomal separation, which deals with crossing over, as well as things like there's 2 laws that we haven't learned about that these demonstrate: the law of segregation, law of independent assortment. We'll get into more details of those later, but it's a good intro to them. And then we have the video where I talk about what sexual reproduction is and why it's important. Other resources you should probably check out would be the Amoeba Sisters Meiosis video. The Amoeba Sisters videos are super duper cute, but they also have a lot of great information and they break it down really nicely. So I definitely recommend you go check that one out. Khan Academy has a nice video explaining what's going on in crossing over to kind of, again, illustrate what is actually occurring at that point. And then we have another Amoeba Sisters video comparing and contrasting asexual versus sexual reproduction. Just kind of letting you know what it is, what's going on, all that fun stuff. All right, so that's the lesson. Again, definitely go watch some of those videos. Consider that part of your lesson. Pick at least 2 of them to go and view to add to your understanding of these different processes. In the meantime, I hope you guys are doing well. I hope you're staying healthy. And I hope that this helps you to understand what meiosis is and why it's important. Now, should you have any additional questions, feel free to email me or hit me up in our Google Classroom. Don't forget, in this time of you can always ask me questions in this crazy time, but if you don't ask your question, I don't know how to help you. So please make sure you do ask questions. Let me know if you're confused or just want to kind of review something. I'm here for you guys. So until next time, take care, you guys, and stay well. All right. Take care. Bye-bye.